Thanks, Dr. Lowery, for that wonderful introduction. Like you said, my name is Nick Weinstein. I'm currently the second year of the BMS program. And my work in Dr. Lowery's lab is with TAS1R receptor proteins and how they affect bone remodeling. Before I start on anything about my research, a little background on the TAS1R receptors. They're a family of G protein coupled receptors that are normally found on taste buds, hence the name. However, recent literature has found that they are also located in the GI tract and skeletal muscle and cardiac muscle, but their effect on skeletal cells has never really been documented. So we began to look into this because there's a paper published in 2014 by Simon et al. Out of the University of Michigan that incidentally found that knocking out the TAS1R3 receptor leads to increased bone mass. They really didn't go any further into this, mostly because it's outside the scope of what they were researching. They just simply noted there was an incidental finding and left it blank. So our lab decided to pick it up and see exactly what the mechanism was behind the knockout model having a higher bone mass. Our first step was to corroborate the findings of Simon et al. As you can see on the right, we have wild type versus knockout mice micro-CTs. And just subjectively, you can tell that the knockout mouse has a much thicker cortical bone. These are 20-week-old male femurs. And then we also quantified it on the left. And you can see that the cortical thickness is significantly higher in the knockout model versus the wild type. The next step was to see whether or not this was an osteoblastic model or an osteoclastic model. In other words, is the bone mass increased because bone formation is increased or because bone resorption is decreased? Look at this, we looked at two markers, P1NP and CTX. P1NP is the hallmark bone formation marker. CTX is the hallmark bone resorption marker. What we found was that in the knockout model, the P1NP concentration went up very, very slightly, nothing significant, nothing there to report. However, CTX significantly dropped, which indicated to us that the resulting increased bone mass is because of decreased bone resorption. So this led us to conclude that the data shows that TAS1R3 signaling in osteoclasts is a key regulator in bone resorption, and it led us to the working hypothesis that TAS1R3 activity and the loss of TAS1R3 activity leads to reduced osteoclast differentiation and or function. The last part of that's important because we haven't been able to, to this point, we did not determine whether or not the decrease in osteoclast activity is because differentiation is different, i.e. it doesn't, the osteoclasts aren't forming, the population is lower, or because they're just not functioning as well. So that was our next step. We wanted to look at osteoclast differentiation first. To do this, we looked at histomorphometry, which quite simply is cutting bones into very, very thin slices, mounting them on slides, and then staining them. And after you stain them, you can actually count the amount of cells on that particular slice of bone. So the two important numbers for our study are the cortical osteoclast number and cortical osteoclast surface. As you can see, between the TAS1R3 knockout and the wild type, there really wasn't any difference in the number or in the osteoclast surface. Also, trabecular parameters were also unchanged. What this told us was that differentiation of osteoclasts in the knockout is pretty much going the same rate as the wild type differentiation. This data was supported by both rank ligand and trap, serum rank ligand and serum trap numbers. The TAS1R3 knockout and the wild type, again, there's really no difference there between those two. So the next step, now that we have essentially eliminated differentiation as a problem, we need to look at osteoclast function. To do this, we used what are called raw 264.7 cells. These are an osteogenic precursor, a very commonly used, widely accepted model for osteoclast functional assays. The first thing we need to do is we needed to be able to prove that the raw cells were differentiating into osteoclasts. We were able to do that. As you can see, the multinucleation here is an osteoclast. The rest of the cells are so the raw cells, but they eventually do differentiate. But this was the best. This was the best picture of a multinucleated osteoclast cell. They multinucleate in the presence of rank ligand. As you can see on the left, CTSK, this is mRNA data of CTSK, which is a marker for osteoclast differentiation. It skyrockets in the presence of rank ligand, which means that the rank ligand is responsible for the differentiation. Another interesting thing to note is that before and after differentiation, TASMR3 protein also doesn't really change. The receptor is going to be there whether or not the differentiation occurs. So now that we know that it's a functional phenotype, we know that the receptor is present, we know that we can now differentiate the cells, we need to look at the function of the receptor itself. To do this, we looked at two very common downstream effectors of TASMR3. That's ERK1/2 and S6 kinase. These are both activated through phosphorylation events. So really, we were looking at phospho-ERK1/2 and phospho-S6 kinase. We wanted to look at these after activation with what's called saccharin. Saccharin is a sweet molecule imitator. It's an agonist for TASMR3. And what we found was that after activation with saccharin, both phospho-ERK1/2 and phospho-S6 kinase significantly increase, which simply means that the pathway is activating in the manner that we expected it to. So going forward, and more importantly, we now need to be able to show that we can inhibit the pathway, because that's going to be the actual model that shows that a knockout of the receptor is causing the increased bone mass. And to inhibit the receptor, we use something called gramarin. It's a widely known antagonist for TASMR3. And while we're still doing some studies on it, preliminarily, we can say that gramarin seems to be able to inhibit TASMR receptor signaling in response to saccharin in the differentiated raw cells. So early on, gramarin is looking good as an inhibitor, but we still need to do some more in vitro studies with that. So going forward, one of the things I'm going to look to do is use what's called a bone chip assay, which is exactly what it sounds like. It's actually human bone chips in wells, and what we can do is we can apply this, the differentiated raw cells to these bone chips, either in the presence of gramarin, in the presence of saccharin, in the presence of both, and we can see exactly how much resorption is occurring. How we'll do this is we'll then stain after a week or so. We can then stain the resorption pits that form because of the cells, and we can quantify the amount of resorption that's occurring in the presence of saccharin or in the presence of gramarin. Something else that we're doing is we're looking to identify a signaling network. To do this, we recently got back some data from an antibody array. What this was able to do was it gave us a multitude of different protein targets, and it essentially told us what proteins are more abundant or less abundant in the TASMR3 network. We used GAPDH as a housekeeping gene and essentially compared everything to GAPDH. So what this is going to be able to do is it gives us more targets to look at as far as the functional effect of the receptor, as well as gives a preliminary outline for a transcriptional network. So again, like I said, just the next steps are confirming transcriptional regulation using saccharin and gramarin, and identifying a transcriptional network, which will help lead us to a better understanding of the TAS1R3 pathway. So I'd like to acknowledge Dr. Lowery, obviously. He is the one that I'm working with in the lab. I'd also like to acknowledge Krista Jackson. She is the one who actually did a lot of the antibody array work, and really keeps me sane in the lab because lab work is tedious. I'd also like to acknowledge Lillian Plotkin and Hannah Davis at the IU School of Med. They were a massive help during the histomorphometry studies. Aaron Brogy and Justin Nathan at Carleton College, they did a lot of the mRNA data. I'd also like to acknowledge late John Martin and the MU Com Faculty Research Award for funding.